very much for inviting me back again. I love coming out here. It's such a great experience, and this crowd is very wonderful, and I love the questions that you guys all give to me afterwards. So we'll try to keep a condensed presentation tonight and leave lots of times for question. And thank you, as always, to Bill Park and the Park family for arranging this and inviting us out every year, because we are certainly very passionate about the work that we're doing and very dedicated to trying to treat this very challenging disease. Because, you know, I show this slide, I think almost every year, because just to remind us about the challenge that we're facing with this tumor, and the fact that glioblastomas in the landscape of cancer are a fairly rare kind of cancer. And the treatment usually involves surgery, radiation, and chemotherapy. And despite this very aggressive therapy, the median overall survival is still fairly dismal at only about 16 months. We have made some advances with looking at different molecular subtypes to these tumors, and so there is a bit of variability in how tumors respond to treatment and their prognosis. But we still have ways to go, because as you can see, since 2005, we still only have 2 drugs that have been approved, although recently there was one device that has been FDA approved for these tumors. And so really very little has changed in the last 12 years, and so Dr. Arriaga and I have our work cut out for us. So, you know, a lot of people are talking about immunotherapy for cancer in general, and particularly for brain tumors. And unfortunately, while there have been some home runs for cancers like melanoma and lung cancer, immunotherapy really has not panned out for glioblastoma. Last year I had said, you know, these clinical trials were ongoing and the results are pending, but unfortunately this year I have to substitute, but the results are disappointing. So really immunotherapy has not panned out very well for glioblastoma. There's still trials that are ongoing, but we need better ways of understanding what is happening in the context of immunotherapy or even other novel agents in general. So I'll talk a little bit about the trial that we're trying to do with the support from Miles for Mary to try to understand what is the role of the immune system in brain tumors and glioblastoma in general. So the trial that we've been doing for these patients really is looking at both MRI scans and PET scans to try to figure out how tumors are responding to immunotherapy and what is the role of the immune system. And the challenge is with brain tumors, it's really hard to go in, take a piece out of the tumor, and look at it under the microscope to figure out how a tumor is responding to a drug that we're giving to it. You know, a lot of patients don't like to go back for serial brain biopsies to understand what's happening. So we're left with other tools such as imaging to try to figure out what's happening. So in this trial, patients are getting immunotherapy, and they're getting brain MRI scans before immunotherapy and then after immunotherapy. And the challenge is that oftentimes their brain MRI looks worse after their immunotherapy. They have more uptake of this contrast dye here. The challenge is it's hard to interpret what that uptake of the contrast dye means. Is that actual tumor that's growing, or is that inflammation from the drug doing what you want it to do? We can't tell from just this MRI scan. Obviously, if the tumor is responding, you want to keep them on that immunotherapy. But if their tumor is progressing, you want to change therapy and do something different. So what we're doing with your support is adding in these PET scans, which are supposed to help us understand the response of the tumor to immunotherapy. Now, this challenge of interpreting our MRI scans doesn't happen just in the context of immunotherapy. It also actually happens even in the context of standard treatment with that chemotherapy and radiation that I was talking about. So even after standard treatment, you can see this increase in contrast enhancement. And again, we don't understand if that represents just an inflammation, a response that we want from our chemotherapy and radiation, or if it's actual true tumor progression. So in this part of the study, we're doing, again, our PET scans to try to measure tumor inflammation and try to figure out what is happening. So where are we now at this point with this clinical trial? We have 10 patients enrolled, and that was our goal, was actually to get 10 patients because this is a pilot study. This PET tracer is a novel tracer that is supposed to try to look at inflammation in tumors. So we're very happy that we've reached our goal of 10. We might actually try to see if we can get a few more patients because the data has been a little bit mixed, and I'll show you that in a minute. And so we might try to continue to enroll a few more patients to better understand what the response is that we're assessing. And we'll be presenting this data at the Society for Neuro-Oncology meeting in November, which is our big neuro-oncology meeting that we have every year. And so I'll show you some of the data that we've found in this study, which unfortunately is not projecting very well. But in this far left panel, you see this is the standard MRI scan, and you see this increase in uptake of the contrast dye right here. This middle panel is, maybe I'll come over here. You can't see this here, but imagine you see this dark area here as opposed to this brighter area over here. And this is the PET scan here, and you see no bright areas on the PET scan. And this actually, patient went on to have, the concern here was this tumor or was this actually inflammation? This patient went on to have a biopsy, and it actually turned out to be all inflammation, and that corresponded to the lack of uptake on our PET scan. Now, this was another patient that we treated who got treated on that immunotherapy arm, and so what you see prior to treatment is that bright white area showing increase in contrast enhancement. There's actually also increase in the blood flow to the tumor and a bright area on the PET scan. After 1 month of treatment, you still see that bright area, but you see this new area appearing over here, and it corresponds to this area over here of INTEEK. And you also see this bright area on the PET scan. And so that suggested that this tumor is actually growing and not really responding to our immunotherapy. So this trial has been a little bit challenging because, to be honest, we thought that our PET tracer was actually measuring inflammation, but it turns out that our PET tracer is more specific for actual tumor growth, which is a little bit counterintuitive to our initial hypothesis. But this is what happens in science. It sort of happens in these fits and starts, and you learn a lot along the way, and you have to be able to rethink your hypothesis and make changes and adapt. And so you always have to have an eye towards what's the next opportunity. And so I've come around to the fact that I'm not sure that this PET tracer is going to answer all the questions that we had hoped for it, but we've learned a lot along the way that we do seem to be able to measure changes in tumor biology with our MRI scans at least. So what's our next step? What are we working on now? So right now we have an ongoing study of a drug that's called bavdegalutamide. And so this is a phase 2 trial that I'm running, looking at patients with newly diagnosed glioblastoma. And bavdegalutamide is a really interesting agent because it activates the immune system in a different way, and it also is supposed to target the tumor cells itself. So we're getting both immune system activation as well as direct targeting of the tumor. And this trial is being funded through the National Comprehensive Clinical Trial Network. And so this is sort of our next step where we're moving our immunotherapy work forward into trying to look at a different approach to targeting tumors. So a lot that we're doing with your support and a lot we're learning along the way. And so now I'll turn it over to Dr. Arijaga.
Dr. A.
She is running our early phase clinical trial program at MGH, kind of bringing new drugs into the clinic for patients with brain tumors.
Thank you. So, so these are slow, and I apologize. I'm going to actually put them all together, but We'll start from the beginning. So I wanted to talk a little bit, so as Elizabeth mentioned, Dr. Gerstner mentioned, I am focused on the development of early phase clinical trials, so getting these ideas and getting them into the clinic. And ultimately, I wanted to talk to you a little bit about, I'll talk a little bit about the end about one trial that I'm involved with Dr. Gerstner, but also about what is a clinical trial, how do we actually get these things running, and actually some of the challenges that we face so that you better understand all the work that goes into what we do and how we can learn from challenges and mistakes to make things better. So this is a slide that will talk about how we actually start thinking about trials. So first, as you see here, we have an idea. Typically, you will have preliminary data, so that's where basic science comes in. And there's animal models for these diseases, and as I'll mention, unfortunately, these animal models aren't perfect, but this is where it all starts. So we have an idea based on basic science, then we have to worry about the funding. So you may have an idea, a great clinical trial, but who's going to fund it? Some of these, the funding will actually come directly from pharmaceutical companies. You might actually have an idea on a drug that they already have. At times they've actually shelved it because they thought, hey, we tried it in one disease, but it didn't work. So you go to them, you ask, or you apply for more government sponsor funding like what Dr. Gerstner was just describing, or through the NCI. Then you have to write a protocol. The protocol, believe it or not, is a very, very long and detailed document, oftentimes over 200 pages, where it really goes into all of the details of how you want to run the trial. And this is critical because you're going to be sending this protocol off to, ideally, if it's a multi-center trial, to lots of different hospitals. And having the protocol be as consistent as possible means you'll get a trial that's as consistent as you want it to be, with results that will be most meaningful. So you generate that, then you submit to your disease committee. So each committee in each hospital or each disease site in each hospital will have a separate committee that has to kind of say yay or nay based on your idea. So that's kind of one of the hurdles. Then if this is a trial of a drug that hasn't necessarily been used in this indication, then you have to file an IND with the FDA, which is for a new drug to be able to be tested in the way that you want to test it in your disease site. So that also has to be reviewed by the FDA, which takes a lot of work and time as well, which I will go into. Then in our own hospital, so lots of different pieces and moving parts, in our own hospital we have to have it reviewed as well. So your disease committee says, yes, you can go ahead with your concept, I like it. Then within the hospital you have both a scientific review committee and an IRB, institutional review board. The first step is the scientific review committee. This is made up of physicians like ourselves that volunteer our time to be able to review these different protocols, and it's quite time consuming as well. Then once the scientific review committee says, yes, it looks good, then you go to the institutional review board, and each hospital has these. There are some that are more central, so for multi-center trials, you could have potentially a central IRB, and then it's easy to disperse to other hospitals, but a lot of the time it actually just goes to the different IRBs in each hospital. So again, fairly time consuming. And then I would say about a year and a half later, you potentially have a protocol that's open. So there's a lot of steps to getting this up and running. Ultimately, you will register for clinicaltrials.gov. For those of you unfortunate to have any patients that may have required trials, you may be familiar with this website because every clinical trial in the United States will be listed there. It's a requirement, so it's a way to kind of look to see what's happening in the nation. And then lastly, lots of reports are forthcoming after you open the trial. So to the IRB, to the sponsor, to the FDA, there's lots of regulatory work, both at the hospital level, but also at the FDA more national level as well. So I wanted to talk a little bit about trials again and let you know kind of what are our goals. So there are three or four, usually four phases, which I'll go into details about, but really they're arbitrary designations in terms of what goals fit into what phase, though we have organized it in such a way. So really when we have a new drug and we want to test it in our patient populations, we have to figure out what's the dose. What is the best dose that potentially will work? And this has to do mostly with, do we think that this is the dose that gets to our target and does the effect that we want, and also the safety. So we 100% need to know both of those, the relationship between how toxic is this and how effective is it being. We also want to know not just a dose, but is it working? So it might be doing what we're thinking it's doing in the cells, but is it actually having an effect on our patients? How does it compare to other drugs? So this drug may work, but maybe it's more toxic than what we currently already have and only provides a tiny bit of benefit. We certainly need to compare to what's out there currently. And then what is the population that's most likely to be benefited? And this is very important, in particular in our disease, because our disease is actually quite heterogeneous. What we mean by that is that even within the tumor, there's different aspects of the tumor that may respond differently to different agents. And then across tumor, so within people again may have different mutations and respond differently to treatment. So it may be helpful, or it is 100% helpful to know that a drug has activity. But even sometimes when we look across everybody, if it doesn't have activity, we still need to be doing a good job to figure out, well, maybe there was a subset of people in here that actually did respond. So understanding in the responders perhaps what makes them responders, which is part of what Dr. Gerstner works with her imaging, is also equivalently important. So how we get this information is somewhat variable, how we test for it, how we do the dynamic pharmacodynamic studies, how we do the correlatives, that aspect of knowing who's responding or not. And then importantly though, clinical trials is really still the mainstay of how we go from having an idea, testing it in animals, to really getting a drug approved. So there's really a long regulatory process and a method by which we go about doing this. So these are the different stages, as I promised, that we discussed just quickly. So the phase 1, and again, these are arbitrary. As long as you get safety, then it's important, but we have definitely bucketed them into different phases, and we typically run trials in this manner. So phase 1 is the first type of phase when we're looking at a drug. It's typically it's really used to learn the pharmacology in humans, and it's typically the first time that the drug goes into human beings. The sample size, so the number of patients that go in the trials, is variable, but somewhere between 20 and 50. The main goal, although some phase 1 trials will also look to see if there's a signal of efficacy, but the main purpose, and that for which the statistics are written, is really just to determine safety. So you need to find out how toxic this drug is, and you need to find out what dose is the maximum tolerated dose. That's a term that you'll hear a lot about. So that will be the dose that will be then tested in phase 2. So those are the goals of the phase 1. Once you get your dose, you can move on to phase 2. Here, you can think of it as a therapeutic exploratory, where the aim is really to estimate, and I stress that importantly, estimate the activity of an agent. And also, because we haven't done lots of people in phase 1, to continue exploring safety, because that will always be something that we have to continue exploring. So this is typically anywhere between 50, 100, some other trials maybe 200. It's variable how these are done. Some of them are single arm, some of them can be randomized. But really you're just trying to see, do we want to move ahead and spend the amount of effort and money that is required for a large phase 3, which is where you would move to next. So the phase 3 is really the confirmatory. So here you want to confirm therapeutic activity. The sizes in terms of sample size are much larger, 500 to 1,000. And you are almost always, this will be a randomized, usually placebo-controlled, if not placebo, but at least controlled to the standard of care. And it will try to see how much activity truly there is in a large patient population. Because it's a large patient population, we never forget about toxicity or safety, so we're still looking at that as well. And then from there, if there looks to be a positive response, a drug may get approved by the FDA. That doesn't mean that there aren't ongoing trials on the drug, in particular because now you have a whole population of patients potentially testing the drug. So there remain to be long-term safety testing in phase 4, which are termed the post-marketing studies. So testing continues for a while. So I said that I would be talking about how expensive and lengthy this process is. In terms of expense, I don't have great data on this, but I'll tell you it's quite expensive. But I just wanted to really highlight here kind of the number of years that it can actually take. So I talked about just at the individual level of running one trial where it could take 2 years to get it open, then add another 2 years to run the trial, then add another year to analyze the data. But when you look at it from the whole research and development scheme, it could take up to 16 or plus years to actually have an idea, then to get a drug into patients. Now, there are accelerated approvals when drugs look like they are particularly promising, and in particular in diseases where there really aren't cures. This can be accelerated, but this is in general how long it takes. So I wanted to talk about some challenges, as I mentioned. These are challenges, really kind of generalized challenges for oncology in general. So one of the challenges that when I think about how we run trials is we're oftentimes testing drugs alone, at least initially. And it's hard to then know once we don't get a signal, say, we'll put it in the back burner, we'll scrap it, when the reality is, there's lots of resistance mechanisms. You might be targeting something, but something else pops up. So knowing that a lot of the time combinations are needed and that understanding what combinations are needed is important would certainly help in terms of these trials. So by understanding, I mean actually doing what we call pharmacodynamic testing. So giving the drug and then actually having access to the tumor. Saying, okay, so what happened? Why didn't this work? Did it get in there? Did a resistance mechanism pop up? Understanding that better is key, and unfortunately we don't do it all the time. So I mentioned this a little bit already, but in every trial, the protocol is just, there's so much regulatory work. So the oversight that needs to occur really, really is tremendous. So there's an average of, for phase 3 trials, an average about 36 administrators who are really looking at this. Just the registration and the oversight of these studies is really quite tremendous. This is another important point, number 3 here. So only 4% of adult cancer patients are thought to enter trials. You would think that it would be more, and that may be in part because hesitancy of patients, but also the availability of trials in certain areas. And this is key to us learning. So while we can learn a lot actually in the basic science and the preclinical models, our models are just not perfect. And in fact, I would say they're far from perfect. So the fact that not that many patients participate is somewhat of a challenge as well. And knowing how many trials there are. So if you think 4% of adult cancer patients participate, there's also a scarcity of patients because there's actually a number of very good trials, and it's hard to get enough patients for each of the diverse opportunities. And then the last point I made here is there's a lot with targeted agents. So we certainly try to get patients who have a specific mutation to go on a trial that has an agent. But the reality is, and we've had some success with that, but the reality is that it's really a small percentage of patients that will have certain mutations for certain that are targetable by certain drugs. So in terms of oncology specifically, so what are our challenges that we face? And I would say one of the biggest ones is, I've mentioned this a few times, but the preclinical models. For us, the clinical models are certainly haven't panned out as as successful as we would have liked. We see lots of success stories in mice, and then when we put it in patients, it just doesn't translate to the same thing. The other thing I will mention here is early phase clinical trial designs. I'll go through this a little bit more, but they do tend to overestimate what the success rate will ultimately be. And I'll explain why we think that is. But if getting these trials to be able to estimate a little better if it's going to be successful or not will certainly help, because you cut out a lot of that time and effort that was spent, and also the precious patients that are enrolling into future trials that we may have been able to know ahead of time were going to be unsuccessful. Very important to us as well, and I've talked a lot about the importance of having tumor to understand why drugs don't work. So for brain tumors, as you can imagine, it's not an easy ask. can I please give you my drug, and can I then go get your tumor afterwards? Certainly, it's actually not something we don't typically do. We sometimes do have access to tumor. This is in patients who are planned to get a resection. So it's not uncommon that there will be a recurrence, and we feel that taking out as much of the tumor again will be clinically beneficial. And in those cases, we will try to enroll them in a surgical clinical trial where they get the drug beforehand, then they go to their planned surgery, and we can learn about the biology. But this is really a fraction of patients. So there is still a lot that we don't get to learn about the tumor biology by not having access to it, basically because it's in the brain. There's some unique aspects and characteristics of GBM that I think also are challenging, in particular to our disease. So there's a lot of abnormal blood vessels. You may have all heard about, the GBM is quite vascular. And the thinking is that there's necrosis associated with this vasculature and may actually lead to heterogeneous drug distribution. So even in studies that we do where we may be able to get a piece of the tumor, we don't really even see is it getting throughout the whole tumor. So that's a challenge as well. And then lastly, you probably have all heard about the blood-brain barrier. This is really, something that evolutionarily was created, which is a good thing. It allows for us to prevent toxins from getting into the brain, which is, such an important site, such an important organ. What occurs is that there's these tight junctions that don't allow for things to pass, and when they pass, there's also these pumps that, that basically pump them back out. So this means that for us, drug development is quite challenging because it has to be drugs that we know cross this blood-brain barrier. That's another area that certainly poses a challenge. And don't forget, we're a small pie of all of cancer. So a lot of the time we take drugs from other cancers that have already been developed, but those drugs are not created with us in mind. So a lot of them, we don't know if they cross the blood-brain barrier, and they're certainly oftentimes not designated to cross the blood-brain barrier. The thinking actually is if it crosses the blood-brain barrier, it might cause more neuro side effects. So oftentimes they're actually created not to. So I just wanted to show you this slide. This is actually a summary of the correlation between response rates at the bottom of phase 2 trials and response rates of phase 3 trials. So as you see, most of these dots are below this line. So what does that mean? In phase 2, they were positive, and in phase 3, they were negative. So this is what I was mentioning about, unfortunately, our early phase trials oftentimes overestimate the likelihood of success, which is something that we have to work on. This is a chart of different phase 3 trials in neuro-oncology. I won't go through all of them, but I just wanted you to see it was recently published in 2017. And these are trials that did well in phase 2 and failed at phase 3 for numerous reasons. Most of you may have heard of bevacizumab. It is still something that we oftentimes will use. This is Avastin in our patients. But the reality is that that trial at phase 3 did not show an overall survival. So a lot of the reasons of the limitations of the phase 2 studies have to do with what we're measuring, how we're measuring. Imaging comes into it a lot with Dr. Gerstner, is working on fixing. And a couple of other things, which I think I go into some of these right now. So, some solutions that we can think of to help fix how we run trials. So I think one of the main things we have to focus on is really the preclinical phase and developing a very strong hypothesis, which is GBM specific. So I mention this because, as I told you, there's lots of drugs that we take that are not GBM specific from its creation onward. Improvement of preclinical models. Again, in the mice, we are not that bad at curing this tumor. But in the humans, it's a completely different story. And then improvement in early phase trials. So one of the things I'll underscore here is randomization. As I mentioned, in phase 3 trials, when we really want to see if there was a response, the gold standard is to randomize patients because it reduces the bias. Some patients get the drug, other patients either get just standard of care or get even placebo. Phase 2 trials oftentimes and historically are single-armed, meaning everyone gets it. And we compare to historical controls. But as we learn more and more, the reality is our historical control survival is also improving a little bit just by us being better practitioners and understanding more of the needs of patients. So that isn't always a good benchmark. So there has been a move to moving more toward randomization of these trials. If you do have single-arm studies, we need to have better endpoints. We oftentimes, the endpoint, meaning is this positive or negative, based on an outcome. Oftentimes, it's the survival, as I mentioned, compared to historical controls, which isn't ideal. We need to try to get more of these studies where patients will be treated before they're planned for surgery so that we understand the biology of what's happening when you get treated in the tumor. Why is it failing? Is it getting in? Is there resistance mechanisms that are popping up? There's this new type of trial that's becoming more popular called platform or basket trials. They're very interesting. We have one that's open in our center right now. And instead of having one arm compared to a single control arm, they'll have numerous arms and they'll have one control arm. So everything's compared to only one control arm. You can imagine as a family member of a patient or a patient that randomized trials aren't, your first choice because you don't know if you're going to get the agent that's being studied. So here, with these basket trials, we limit the number of patients that will go on the placebo or standard of care arm. These trials also try to learn from prior experiences of the patients in the trial even, and then try to put patients in different arms based on characteristics of the patient. So they're becoming more popular. We'll see how helpful they really are to our research, but it's something else that's popping up and that we have great hope and promise for. And then the last thing is understanding the human biology, not necessarily just based on the tumor tissue that hopefully you would get, but just based on different characteristics of the patient and the tumor, even at initial diagnosis. So an example of this would be the imaging that Dr. Gerstner does, understanding why did this patient respond and this one didn't. Was there something that we learned about this patient's imaging suggesting that there was, more penetration or increased hypoxia that meant that maybe this population is going to be the one that will respond to treatment? The same with genetics, as an example, from the initial tumor. Is there something about a specific population? Is this the group that responded and why? So understanding that is also important. So with that in mind, I wanted to talk to you about a phase 2 trial that actually should be open any day now at our institution. It took a while, as I told you, the long timeline of different things that occur. So this is a trial that I'm heading. It's a multi-center trial run through a consortium, the ECTCN. So it will be open nationwide. Dr. Gerstner has been instrumental in the creation of this concept, and then also she's running part of the imaging correlatives for the trial. So this is a phase 2 trial in recurrent GBM. We're randomizing patients, so notice I am randomizing, so as I mentioned, there's pluses and minuses here because there will be some patients, unfortunately, who won't get the treatment. But the hope is that we'll have a true answer. So they're getting randomized to a combination of agents called cediranib and olaparib, and I won't go too much into the biology of this, but I will tell you that this is a combination that has been successful in ovarian cancers, and we do believe that this combination could have some activity in our tumors. That's randomized to Avastin, which is really the only FDA-approved drug for recurrent treatment. There's others that we can try, but in terms of our standard of care, there's no clear one. This is the closest that we can get to is Avastin. And they will continue until progression, or if there is untoward side effects, then they would also stop the trial. So in the bottom here, you see that there's a number of different correlative studies. So we've incorporated lots of different ways to study the tumor biology to try to see if this is a negative trial, which are the patients who responded and why? Can we make some sense of that? So we are incorporating some blood markers, which we think are related to some of the activity of one of the drugs. We're also incorporating some tissue markers from the initial surgery, which we think are also related to possible activity of the second drug. We're looking at the tumors and doing whole exome sequencing, so looking at all of the genetics of the tumor to see if there is some gene that potentially corresponds to activity. And then lastly, we're doing advanced imaging to see if there is any particular aspect of the imaging that can predict who may best respond. And that is all I have, guys. So thank you so much. I'm honored to have been invited. I don't think I said that in the beginning, but this does seem like a wonderful group, and thank you so much for all of your support.
So thank you.
So my question is, can you put some numbers to the phases in the trial? What's considered reasonably acceptable? When can you continue, and when do you have to pull the plug? If you're dealing with a cancer that is essentially going to be fatal, even if there's only a 10% chance of survival, that to me would seem better than zero. But obviously, there are ethical issues here. So can you put numbers to the stages, and how are these decisions made?
There's actually a pretty simple formula, believe it or not, although it is changing somewhat in human trials. Most phase 1 trials, which is where we're looking at toxicity, are arranged based on a 3x3 design. So you treat 3 patients. You have your number of serious adverse events, or you classify, and this is just a general classification, what is considered a serious adverse event, and this is general across oncology, going into the hospital, life-threatening, anything like that is considered serious. If one of your 3 patients has a serious adverse event when you start at 1 dose, then you have to do another 3. If 2 out of 6 have a serious adverse event, then that's it. That's your maximum tolerated dose. If 3 out of the 6 have a serious adverse event, you have to push your dose down. So there's actually been a pretty standard way that we do this. There are different ways to do it, but kind of to give an answer to what you're saying. The reality is it's very tough, for both of us. We take care of patients too. And I say this a lot to my patients. If I could cure you, I would put you through the ringer and back and all toxicities, and at least there's a hope that I can cure you. Quality of life is a big, big component of that. And that's why it becomes hard. It's very hard. I mean, yes, you hear about the war on cancer, and in the 70s and 80s, they were doing this. They were killing some kids as they were trying to treat lymphomas. And you know what? They did get a lot of answers. So there is, a back and forth. There's a balance. But I think that when it's your individual family member or it's you, it's very hard to say, hey, I sacrificed myself for that war. You want the last year that you have to be as good as you can make it, and that is really our pledge to our patients as well.
So what you pick as your toxicity and your level of acceptance of toxicity does depend a little bit on the stakes. And so you can be selective in what you say is a toxicity that will make you change your dose for the next cohort or reduce your dose. So you do have a little bit of judgment as an individual running the trial as to what you accept as toxicity or not. But as Isabel was saying, it's really a balance between, not causing more harm than good. And there was this concept that you would push everything to the maximum tolerated dose, because we thought, as Isabel was saying, if you flood somebody with chemotherapy, that was the way to get rid of the cancer. But we're finding out that there's this new concept of the optimal biological dose. You don't necessarily need to push a dose to, that maximum dose that the human being can survive. You might actually want to figure out what is the dose that's the optimal biological dose. What's the dose that's most active for a particular type of cancer? Can you kill tumor cells without also harming the patient? And that's where those correlative studies come in to help you figure out, are you reaching your target or not without causing more harm than good. So to make a long story short, there's a lot of thought that goes into how you balance toxicity with outcome.
I'm sorry. Is there a redundancy in clinical trials throughout the country? Are different hospitals or hospitals throughout the country looking at the same data, trying the same drugs, or is it segregated to, like, Mass General's doing this, and other hospitals throughout the country are doing other exploratory trials?
There's variability in that response. I mean, if you have a drug company sponsored trial, they tend to know where their drugs are, and they don't tend to do redundant trials across multiple sites, or they try to make it a multi-center trial. So we have the trial open here, they have it open at Sloan Kettering in New York or MD Anderson, and so it'll be the same trial but open at multiple sites. So there tends not to be a lot of redundancy. The other thing is if it's a consortium-funded study, like the trial that Isabel's doing, the ECTCN, again, it's open. It's an NIH-sponsored consortium, but multiple different sites can participate, which is very important for a rare cancer like ours, so that the trial is available to multiple people across the country. So there's not a whole lot of redundancy. You know, there's not another person who's running a bavdetuximab trial for GBM at some other center.
Is the data shared throughout the country on all these trials?
So there's a big push to share data, so that clinicaltrials.gov website that Isabel mentioned, we are supposed to submit our data to clinicaltrials.gov within X amount of time of the study being published and the results being released. It's a very cumbersome way of sharing the data. If you participate in a consortium like the ECTCN or the Adult Brain Tumor Consortium or the Alliance or these other NIH-funded consortiums running multi-center trials, they're also supposed to share their data because they're taxpayer-supported trials. And so you can petition those consortiums and say, I would like access to one of the big, large, randomized GBM studies that I've done, so I can try to dig into the data and figure out, if I look at it in a different way, can I identify a signal in a subpopulation? It's easier said than done, but there is a push to try to do that. Pharma makes it a little bit more challenging, but the NIH is really making a big push to try to share data across different investigators. If you don't update your clinicaltrial.gov website with results, the government will fine you. And so it's something like a $10,000 fine per day. That's a little bit of an exaggeration, but it's a pretty hefty fine that they will fine you if you do not update your results. So MGH now has a whole group of people who are helping support us with clinicaltrials.gov.
There's some people around here that I talk to who feel like there are more glioblastomas in this general area than ever. Do you see any trends in diagnosis or treatment?
If you look at the national trends and the national statistics, there really hasn't been a change over the last several years that we have statistics for. It's still only about, 8% of patients, and that's been pretty stable over the last 5 years that we have data for. And if you want to hone down on Massachusetts individually, I don't think we have a sense from our volume at MGH that there's been a big change, but it's a little bit harder to tease out specifically in Massachusetts what the incidence rate is.
Yeah, and I agree. From clinical practice, at least, it hasn't seemed to have been increased in comparison to other years. I do think that having it in the news more, being more publicized, people are speaking about it more. And it used to be a disease that not that many people knew about, but is now more in the limelight. Certainly, I get questions about this all the time since I'm, being a neuro-oncologist, I feel like it's quite prevalent, but when you really look at the statistics, then it hasn't actually changed too much.
Any of this information is shared on a more formal basis internationally, and is our clinical trial in the U.S. process much more stringent? I believe it is, but I'd like you to comment on that. The other question I have is on the clinical trials. It seems to me you said early on that there was a very small percentage of patients. Is there an attempt to increase that, and would that make a significant difference?
Germany has very little challenge in enrolling patients in clinical trial for brain tumors. And I think there's just something about their community that encourages people to enroll, and they sort of say, here's your clinical trial option, let's have you participate. There just seems to be a different attitude towards trial. I don't know if it's a more paternalistic society or what. Here we're sort of very much into shared decision making. And there are a lot of regulatory hurdles that need to happen to get patients on trials. But the data that we generate here, all the data that's on clinicaltrials.gov is available to anybody in the world. And there is a push to do multinational trials. There are lots of regulatory hurdles around that, but there certainly is interest to do multinational trials. So I think it's a little bit harder to get trials up and running here. Maybe
Well, I was just going to say a couple other points. In terms of data sharing, I guess one of the things that the way we also share our results is often through these conferences, and there's lots of national conferences. In fact, there's a European Society, ENO, of Neuro-Oncology, which is in Europe, which a lot of us will go to. Our conference that's actually later this month here in the United States, it is full of people from all around the world. There is certainly dialogue and knowledge that is spread at these conferences. The other point I will mention is that I agree that maybe we're a little bit more stringent in some of our regulatory, hurdles. But I will mention the case of bevacizumab, Avastin. So Avastin was FDA approved here in the United States and ultimately was not approved in Europe because it didn't pass the regulatory kind of red tape that they had. Also, in the running of the trials, maybe we are a little more stringent, but maybe there in the how much you're going to pay and what you're going to pay and how expensive this is and how much rigorous data you need to have to prove it. Maybe it's a little bit off, or it might just be a case-by-case basis, but it's not across the board that we are more stringent. And then the last question you mentioned about the small numbers, the important thing to mention here is that that's across all cancers. And there are a lot of community hospitals throughout the United States. So I would say the availability of patients to be able to go on trial is also low. So it's not just patients saying no. There's also a lot of patients who don't have access to trials, who would have to travel, who have to move to Boston, say, because actually our trials oftentimes require that you get radiation where we are, and that you come for follow-ups, every few weeks. So the trials are not homogeneously spread throughout the United States, and there's a lot of people without access to them as well.
We have people who live in Pittsfield, and they don't want to come to Boston for a clinical trial if it's a weekly visit or even a monthly visit, because nobody wants to drive down 90 and drive into Boston. So there are other hurdles.
Are there any approaches that involve a combination of drugs and some physical aspect, such as actually putting a probe in the middle of the tumor and changing the temperature or changing the pH balance, or other things that are going to perhaps get around the blood-brain barrier problem and enough said, a combination, as I say, that goes beyond just drug therapy?
There's been one device that's been approved for glioblastoma in the last couple of years. It was a positive phase 3 trial showing a mild survival advantage. And so basically what it is it's this hat that you wear, and it's supposed to shoot alternating electrical currents through your head, and it's supposed to disrupt cell division, so stop cells from dividing. The company shows these beautiful pictures of things in petri dishes and the cells stopping to divide, but as Isabel was saying, a petri dish is very different from a human. It's also much easier to get a device approved than a drug approved, because you really just have to show that you're not harming somebody. Now, advocates of this, you can sort of see where my bias is, but advocates of this do show a survival benefit, but there were some flaws with that study. But it is FDA approved and it is an option. So the whole point of this is really to confirm what you're saying, is that there are people who are thinking about this disease in a different way outside of just drug therapy. And so I do think this is an interesting approach. I think it needs to be tweaked. The problem is you're sort of wearing this thing for 18 hours a day and you have to plug yourself into a wall or have a battery pack. So it's a little bit of a quality of life. You have to, it's an investment for the patient. The other approach that people have tried is along the lines of what you're talking about with laser thermal treatments, where basically they'll take a probe and at the time of surgery they'll stick it into the center of the tumor. And this can work for tumors that are hard to get out, meaning that if the surgeon were to go in and try to cut it out, he might leave the patient weak on one side or cognitively impaired, and that's not obviously something we want. We want our patients waking up from surgery looking as good as they do going in. So with this laser probe, they can go in and they can ablate the tumor. They can destroy the tumor from the inside out. And so that's a new technique that we've been exploring and trying, and is showing some promise for those hard to reach tumors. So there are other ways of looking at it. There are other interesting dyes that you can inject, or actually the patient can swallow as a pill before surgery, and the dye goes to the area of the tumor cells. And so when the surgeon is in there looking at the microscope trying to dissect the tumor, this dye lights up under their special microscope, and they can actually be more specific about cutting out the tumor cells themselves and not cutting out normal brain cells. So there's a lot of interesting approaches that are being explored besides just chemotherapy approaches.
You mentioned how you test on animals and mice. How do you find one with the disease in order to test on it and keep it accurate?
When a patient goes for surgery, we take out their tumor, but we keep those tumor cells alive and we keep them growing in the lab. And so you can grow them in a petri dish. And so it's a human-derived cell line from a human tumor. And so then you take those cells and you inject those into the rat, into their brain, and then the tumor starts to grow. So as Isabel was saying, we're trying to improve our preclinical models. And so this is one way, the pipetters have been trying to improve their preclinical models, is making it more, representative of a human tumor itself by using these human-derived tumor cells.
But oftentimes, so one thing you can think about is the environment then in which this tumor is growing, is very different. So oftentimes, actually, we have to have mice who have no immune system, as an example, for the tumors to grow, which is very different. I mean, that is like one of the basic major differences. And we don't have to always do that, but oftentimes we do. And as you can imagine, we're looking into the immune system to see how it can attack cancer. So how is this model, even close to mimicking what we see in humans?
Okay, well, thank you.
Thank you, everyone.